right, so we're going to go ahead and finish up those two scenarios, and I'm going to give you some background information on both of them, just like I did yesterday. So scenario 4, the stem cells in the early embryo are different from any other type of cell. They are immortal, meaning they can grow and divide indefinitely. They are also pluripotent, meaning they can give rise to any type of cell in the body. These powerful characteristics give stem cells enormous potential for curing diseases. Before we can unlock the potential, we must first understand what is happening inside stem cells to make them different from the other cells. We know that as stem cells differentiate into other cell types, many genes are turned on and off. So, an important step in understanding stem cells will be to identify which genes are active in a stem cell and which are not differentiated cells. You are a researcher designing a study that will compare the gene expression in stem cells with the gene expression in differentiated cells. So the only special consideration here is that the stem cells should be easy to obtain and manipulate in a lab. So a little bit of background information about that. Differentiated cells can be reprogrammed to look and act like stem cells from an embryo. In early reprogramming experiments, scientists removed the nucleus from either an egg cell or a human embryonic stem cell and replaced it with the nucleus from a differentiated skin cell. The resulting cells were pluripotent, meaning they could differentiate into many different types of cells. Scientists deduced that egg cells and ES cells contain certain factors that can reprogram a differentiated cell's DNA, bringing it back to an embryonic state. Researchers at Kyoto University wanted to figure out which factors present in eggs and embryos were responsible for reprogramming. They identified 24 genes that they thought might be important and used retroviruses to deliver them into skin cells taken from a mouse tail. Adding these genes made the cells pluripotent. Gradually, they eliminated genes from the mixture until they identified the minimum number of factors, or gene products, required to reprogram a nucleus. They were surprised to find that adding just 4 genes was sufficient to reprogram the nucleus and make the mouse skin cell pluripotent. The same researchers later programmed human skin cells by adding the same 4 genes. Stem cells that are regenerated by reprogramming the nucleus of a somatic cell are now induced pluripotent stem cells, or iPS cells. So you can pause the video there if you are working on your little part there about advantages, disadvantages, and what type of cell you would want to use. Obviously, if you're making the iPS cells, you probably can't use the iPS cells. They wouldn't be the best ones for that. And the last scenario is going to be about lupus. So in a healthy person, specialized cells of the immune system recognize and attack foreign invaders such as bacteria and viruses. Lupus is a disease in which the cells of the immune system become confused and attack the body's own healthy cells as the disease progresses. The immune cells attack the body faster than it can heal, causing severe damage to the patient's joints, skin, kidneys, blood cells, heart, and lungs. In an effort to stop the destruction of healthy tissue, patients take medications to suppress their immune cells, but these medications leave the patient very vulnerable to infection. You're a researcher who is beginning to study stem cell treatments for lupus. One treatment looks promising, involving first destroying the patient's bone marrow, which contains faulty immune cells, and then replacing it with properly functioning genes. Special considerations would be the stem cells should be easy to obtain and manipulate, and the stem cells should be able to differentiate into immune cells. So a little background here on that. Systemic lupus is an autoimmune disease that primarily affects young adult women. Most patients are able to manage the disease with aggressive immunosuppressive medications. However, some patients do not respond to medication, and the disease becomes life-threatening. A stem cell transplant therapy for this subset of patients is now in clinical trials. The purpose of this therapy is to reset the patient's immune system and thereby restore organ function. First, a patient's own blood is removed and treated to enrich for bone marrow stem cells. This will give rise to healthy immune cells. After chemotherapy destroys the remaining blood and immune cells, the enriched stem cells are returned to the patient by intravenous injection. The hope is that the injected stem cells will be able to repopulate a patient's bone marrow and heal the immune system. 48 patients received this treatment. About half were disease-free after 5 years, and no patients died. It is too early to tell whether the therapy cures the disease or simply slows its progression, but scientists hope that it may be a safe, effective alternative for patients whose lupus resists traditional therapies. Interestingly, the same type of stem cell transplant has been used for decades to treat a number of other diseases, including certain forms of cancer. An additional stem cell transplantation therapy for lupus is currently being investigated in mouse models. So you'll want to go through, tell me which type of stem cell you think would be best for being able to differentiate into an immune cell. And then we'll turn that in. And tomorrow you're going to do some questions, some comprehension questions, just based off of the notes that we did earlier in the week. All right, have a great day, guys.